Good morning. Our lecture today is dentinogenesis. Dentinogenesis is the process of dentin formation, which starts before amelogenesis or enamel formation. Dentin is formed by the newly differentiated odontoblast cells, which come from the peripheral cell of dental papilla under the organizing influence of the inner enamel epithelium. Dentinogenesis takes in two phases. The first one is the formation of organic collagen matrix, which is called a predentin, and the second phase is the deposition of hydroxyapatite crystals on this predentin. At the beginning of dentinogenesis, the odontoblast elongate, resulting in the formation of Tom's fibers or odontoblastic process from this odontoblast toward the new dentin formation. Odontoblast then secrete matrix protein at the apical end of the cells and along its odontoblastic process. The secreted matrix is collagenous and not mineralized, hence it is called predentin. As the matrix is being secreted, the odontoblast move toward the center of the future pulp. The matrix that formed around the elongated cell process or odontoblastic process, or also called Tom's fibers, eventually mineralized, and the odontoblastic process will lie within dentinal tubules. So at the beginning, the predentin is unmineralized. Then when it is mineralized, the odontoblastic process, which comes from the cytoplasm of the odontoblast cell, which come inside the dentinal tubules, which is surrounded by a mineralized matrix of dentin. So the dentinogenesis is, as we said, is two processes. First is a matrix formation, or called the predentin, which composed of collagen fibers type 1 and type 3, and the ground substance, which composed of glycoprotein and protoglycan and non-collagenous protein. Second phase is the mineralization of this predentin by addition of hydroxyapatite crystals, which come from the odontoblast itself. As each day passes, predentin is formed along the pulpal boundary. The adjacent predentin that was formed during the previous day mineralized and become mature dentin. So every day is the dentin formation, predentin formation. The previous layer, which is had for hydroxyapatite on it, become a mineralized dentin, and the new layer of a predentin will formed adjacent to the odontoblast. During the period of crown development, approximately 4 micrometer of dentin is laid down in every 24 hours. So daily formation of a predentin is about 4 micron. Incremental deposition and mineralization of dentin begin below the cusp. So the beginning of the dentin formation is start at the cusp tip, and then at the tip of the pulpal horn at the dentino-enamel junction, and then continuous cervically. Dentinogenesis continuous, lifelong, but slow down after the tooth is completely erupted. So the dentinogenesis is continuous throughout the life of the tooth when the pulp is vital, because the odontoblast cells is present in the peripheral side of the pulp. So continuous dentin formation, but this dentin formation is become slow when the tooth is erupted inside the oral cavity. This is the advanced stage tooth germ, and this is the cusp side when the dentin beginning dentin formation after the differentiation of this odontoblast, while this area there is still the odontoblast not differentiated yet, so there is no dentin formation here. So the beginning of dentin is in the cusp area and then continuous cervically till reach this area. And this is the dental lamina, which is disintegrated from the oral epithelium. The differentiation of odontoblast. This picture shows the diagram represent the undifferentiated mesenchymal cells of the dental papilla here, and this is the basement membrane that separated this cells or peripheral cell of dental lamina from the inner enamel epithelial here. So the inner enamel epithelium and dental papilla cell will be histodifferentiated in early pill stage, and then when the differentiated of odontoblast and secreted of peridentin, the stage is called advanced pill stage. So if you focus here on the differentiation of odontoblast, this is undifferentiated, become mitotic division, differentiated from the peripheral dental papilla, which this cell is undifferentiated mesenchymal cells. Then after mitotic division, we have two daughter cells here. One of them is become a new cells, and this remain as stem cells. This begin to as pre-odontoblast, become from the oval shape to the cuboidal, and then columnar in shape. The nucleus is centrally located and the organelles is not fully formed. There's also a basement membrane between these cells and the pre-ameloblast, which is came from the inner enamel epithelium. Then these cells become odontoblast and begin secreted of a new formed layer of peridentin. Also, this cells, the nucleus of these cells become basally located toward the pulp, and also the organelles become well differentiated here, and there is a cytoplasmic process begin toward the newly formed peridentin. And this picture, this is also the odontoblast during secretory stage of dentin. The dentin matrix is become thicker, and also there's a differentiation here of the ameloblast and become secretion of the enamel. So there is a dentino-enamel junction formed instead of the basement membrane between these two cells. This also the odontoblast and the cytoplasmic process become taller and become inside the dentinal tubules, which is a mineralized dentin here. And this cells, this is also the odontoblast, but it's in the quiescent stage when there is a slow formation of dentin after eruption. While during the secretory stage, the odontoblast become very tall, columnar in shape, about 40 micron in length and width about 7 micron in width. Formation of peridentin. This diagram shows the odontoblast during secretion of peridentin. This is the nucleus toward the pulp, as we said, basally located. And this is the organelles, which is rough endoplasmic reticulum and Golgi apparatus and mitochondria present, well defined and seen in ultrastructurally under electron microscope. This indicate this is cells is protein secretory cells. This become the secretion of a first layer of peridentin adjacent to the odontoblast, and also there's a presence of cytoplasmic process or odontoblasmic process come from this odontoblast, also called Tom's fiber. And the first secretion of predentine contain collagen fibers type 3, which is called von Korf's fibers. This fibers is perpendicular on the basement membrane, which separated this odontoblast from the inner enamel epithelium. This fibers, as we said, is perpendicular on the basement membrane, is called von Korf's fibers, which is present in the dentin of the crown, while this fibers present in the dentin of the root is differ, which is parallel to the basement membrane. The fibers of von Korf's fibers present in the newly mineralized dentin is called mantle dentin, while the fibers present in the circumpulpal dentin, which is the take the full thickness of the dentin, which come after mantle dentin. Both mantle and circumpulpal is called primary dentin. The fibers present in circumpulpal dentin is type 1 fibers, not von Korf's fiber, but type 1, and this fiber is parallel to the basement membrane, while the fibers present in mantle dentin is perpendicular and thick fibers and type 3. Also, the cell, odontoblastic cell, there is a tight junction and junctional complex between adjacent cells during secretory stage of odontoblast. And this is the odontoblastic process or Tom's fibers, which are present inside all thickness of the dentin, which is formed inside the mantle dentin and circumpulpal dentin. Both mantle and circumpulpal is called mineralized dentin, while the predentin is unmineralized dentin, which is formed in the beginning, and when it's become mineralized, it's called either mantle or circumpulpal. This histological picture shows the differentiated odontoblast, and this is pulp, and this is the beginning of the secretion of the predentin, a blue color, and these cells become ameloblast once before it is inner enamel epithelium, and adjacent to ameloblast is a stratum intermedium and stellate reticulum, which is other layer present in the enamel organ. Here the odontoblast, while here is still undifferentiated mesenchymal cells of dental pulp. Between the dental pulp and inner enamel epithelium, there is cell-free zone or a cellular zone with a basement membrane, while here is the differentiated odontoblast and beginning of predentin secretion. In this predentin, there is von Korf's fibers here, which is perpendicular on the basement membrane, which separated this from the ameloblast. A predentin, the odontoblasts form the main component of the dentin matrix, which is called predentin. The collagen fibers and mucopolysaccharide present in predentin with non-collagenous protein, which form the ground substance. The main non-collagenous protein present in predentin is bone morphogenic protein 247, dentin phosphoprotein phosphorin, DPP, osteocalcin, osteonectin, and osteopontin, and also dentin sialoprotein. This is main non-collagenous protein present in predentin. This aid in the during mineralization by the hydroxyapatite is attached to the collagen fibers. This aid for this attachment. The odontoblasmic process, which is formed from the cytoplasm of the odontoblast, At the beginning is multi-process, and then is become one odontoplasmic process, which is continuous inside all the dentine thickness from the dentino-enamel junction to the body of the odontoblast cells, which are present inside the pulp. This odontoplasmic process is laid down inside the dentinal tubules, which is present in the mineralized or mature dentine. This is represent the dental papilla cells, which is at the beginning undifferentiated as the oval shape cells. When there's an organizing influence from the inner enamel epithelium, they become odontoblast and begin to secrete the dentin. Also, these histological pictures of the odontoblast. This one, this another, and this is the nucleus toward the pulp, while this is the odontoplasmic process of the cells, which are present inside the dentinal tubules here. The black color is represent the mineralized dentin, while the light color dentin represent the predentine. The predentine is usually adjacent to the odontoblast cell body. This is diagram show the odontoblast body, and this is the nucleus toward the pulp. This body is present on the peripheral side of the pulp, and this is the beginning of the secretion of the predentine, which is lighter here. And this type is the circumpulpal dentin, and this type is the mantle dentin. Both of them is primary dentin and is represent the mineralized dentin, while this is the Tom's fiber, which is present inside. The black color is represent the dentinal tubules, which is present inside the all thickness of the dentin. This represents the dentino-enamel junction, which separate between the dentin and the enamel here. Formation of mantle dentin. After differentiation of odontoblast synthesis, first dentin collagen matrix or predentin. This collagen deposit in ground substance of dental papilla. Appearance of distinct large diameter collagen fibers, which is called von Korf's fiber, is the first sign of dentin formation. So the first sign of dentin formation is the formation of von Korf's fiber here, which formed from this newly differentiated odontoblast. This von Korf's fiber present inside the predentin, which is secreted by odontoblast, and this is present only in the mantle dentin. This fiber is perpendicular on the basement membrane and regarded as type 3 collagen fibers. This histological picture shows the odontoblast here, odontoblast body, which are present in the peripheral side of all the pulp, in the crown and in the root. And this is light color is represent the predentin, which is unmineralized dentin matrix, which contain collagen fibers and non-collagenous protein and ground substance. And this dark color is represent the mineralized dentin and circumpulpal dentin, and when adjacent to the enamel and cementum is called mantle dentin. This represents the dentinal tubules here, and this is the mineralization by globular mineralization of the this predentin by addition of hydroxyapatite crystals, which come from this odontoblast. Hydroxyapatite crystals, the first crystals formed from the odontoblast present inside the bud in the cell membrane of each odontoblast cells. This bud represent matrix vesicle, which contain hydroxyapatite crystal, has calcium and phosphor with alkaline phosphatase. When these hydroxyapatite become larger, they cause the rupture of this matrix vesicles, and this matrix vesicle content is put on inside the peridentin on the collagen fibers, and then aggregation of these hydroxyapatite crystals and form the globule. This is the globule, which is represent the accumulation or aggregation of the hydroxyapatite crystals and diffusion and formation of mineralization, which is called as globular mineralization, which is present inside the mantle dentin, which is the first layer of the mineralized dentin. So the maturation of a peridentin, or the second stage of dentinogenesis, it occurs parallel to the matrix formation. It begins at the tip of the crown because the beginning of a peridentin is laid down in the tip of the crown or cuspal edge. It proceeds in the rhythmic pattern to gradually complete cervically, at the beginning in the tip of the cusp of the crown, and then laid down cervically. The first layer of a peridentin begins its maturation or mineralization in a globular pattern by matrix vesicle. As we said, matrix vesicle is the budding present in the cell membrane of odontoblasts, contain hydroxyapatite crystals and alkaline phosphatase, where small center of calcification spread constantly until they fuse together. These hydroxyapatite crystals fuse together and form a globular pattern. If somewhere those globules do not fuse together, area of uncalcified dentin are called interglobular dentin. The globular dentin appear as light in color, while interglobular dentin appear dark in color. During mineralization, there is two mineralization process, a primary and secondary. Primary by the formation of globular pattern by matrix vesicle, and the secondary phase is by addition of hydroxyapatite crystals and growth of these crystals. In interglobular dentin, there is only the primary mineralization, and there is no fusion of the globules, so the area is called interglobular dentin and appear dark in color and seen during the near the dentino-enamel junction in the crown area. The mineralization, this is the matrix vesicle, which contain the calcification, center of calcification by presence of calcium and phosphate ion undergo crystallization and form hydroxyapatite crystals, and these also contain rich in alkaline phosphatase enzyme. This matrix vesicle, this is the electron microscopical picture of matrix vesicle. When this rupture of matrix vesicle, the mineralization of mantle dentin will begin. This is also the odontoblast layer, and this is the peridentin, and this is the mineralized dentin. This is the dentinal tubules in mineralized dentin, and this is the globular mineralization. This is the cusp tip of the anterior tooth. This represents the mature dentin, dark in color, and light color is represent the peridentin, and this is the odontoblast cells present in the cusp tip, and this is the ameloblast and newly formed enamel here. So there's between the enamel and dentin is the dentino-enamel junction. We have hereditary condition called Dentinogenesis Imperfecta, where is there's deformity in the during mineralization or secretion of the dentin matrix. There is the hereditary condition called Dentinogenesis Imperfecta. We have three types of it. the tooth become blue-brown discoloration of the enamel, pulpous crown with cervical constriction, enamel defect. Enamel is loosely attached and tend to chip away from the dentin easily. January of related pulp chamber and canal. Periapical pathology occur. Very difficult to treat, so the teeth become rapidly worn down to gingiva in severe cases. So these teeth need to be covered by a crown, or maybe lead to extraction and then implant treatment. So this is the deformity in the dentin during dentin matrix formation or during mineralization. That's all for this lecture. Thank you for listening.